So we just finished talking about our different cell types, our eukaryotic cells, our prokaryotic cells. Now we're going to talk about how do those organelles function inside of the cell to allow that cell to function properly. And the first thing we're going to talk about is what actually happens inside of a cell to transport things in and out of it. In order to do that, we need to know or review a couple of terms. One, what is homeostasis? If you look at the word, we have looked at this kind of homeo or that homo word before, and that means the same. So what homeostasis is it's trying to get an environment that is the same or that is stable. So for example, in humans, in order for us to function properly, our temperature needs to be around 98 degrees. Our body, it keeps our temperature at 98 degrees. If we do not, then we fall out of our homeostasis and things do not go right. We get a fever, we don't feel good, and our body tries to reestablish that balance. So another way that we can look at this is calling it an equilibrium. So homeostasis is essentially the process of keeping things equal. And in a cell, we're going to look at keeping the amount of molecules inside the cell the same as the amount outside. Or if there's too many molecules outside, bring some inside so there is this even amount or this homeostasis or equilibrium between the cell and the outside environment. In order to do that, we have to rely on this thing called the cell membrane. If you remember, in an animal cell, that's the very outer layer, and that plant cell, it was the middle layer. This determines what goes in and out of the cell. So it helps to keep this balance or this homeostasis. So essentially, the cell membrane keeps things equal within a cell. So because this is the most important organelle in keeping kind of this homeostasis, allowing things to move in and out, once again, let's look at this function. Remember, we called it selectively permeable. That meant it got to decide what came in, what came out. Not everything got to come in, not everything went out. It was kind of, we called it the bodyguard, or if you compared it to like a club, a bouncer. It determines who comes in and out. It's also going to support, shape, and protect the cell. And then something we didn't talk about, it also communicates with other cells because all the cells need to work together, so they all need to keep this homeostasis or this equilibrium with all the cells. When we look at this layer, it looks like a double layer. There are two layers to it, and this allows it to have a flexible structure, allows the cell to kind of have some movement to it. And then it also forms a really nice barrier between the cell and its surroundings, and this protects the inside of the cell from things that may want to come into the actual cell itself. So looking at the cell, we're going to look at three different structures here, and you can see it kind of looks complex, okay, but we're going to break it down. The first one are the phospholipids, okay, and this makes the actual bilayer or that double layer. So I have one layer here and I have one layer up here. And you can see in the phospholipid, it kind of has something that looks like a head and something that looks like two tails. The next thing are these proteins. Those are going to be these purplish bluish things. These will also help in transport. We will talk about that a little bit later in the notes. And then the last thing are these carbohydrates. And these kind of look like the chains coming out of the proteins. And these will help in other processes. It can help in transport, but these are the three things that are actually making up this cell membrane. So we're going to focus mostly on this phospholipid bilayer. You can see once again it has the head, it has the tails, and there are two layers. That's why we call it a double lipid layer. Remember, lipids are essentially fats, and these are phospholipids. And when you see this bilayer, remember bi means two. So once again, it gives the membrane a flexible structure. It's going to form a really nice barrier between the cell and its surrounding. It's going to protect that cell. And when we look at the actual structure, we're going to look at two parts, the head and then the tails. The head is what we call hydrophilic. So hydro is going to mean water and philic will mean like. So the heads are water-liking or water-loving. They actually are going to attract water to them. Now the tails are the exact opposite. They are hydrophobic. They repel water. Essentially they are water afraid. So they're afraid of water. They do not want the water in here. So if water were to get in there, it would quickly be pushed away from it. So when we look at movement across the plasma membrane or cell transport, we're going to look at two types, passive transport and active transport. When we look at passive transport, energy is not going to be used. In active transport, it has to have energy. It's moving against what the cell wants, so it has to use energy to push it that way. In passive transport, we're going to talk about 3 different types: diffusion, osmosis, and facilitated transport. In active, we will actually talk about 2 types: our protein pumps, and then these things called vesicles that will help move larger molecules. Most specifically, we'll talk about endocytosis and exocytosis. For tonight, we're just going to focus on this passive, and more specifically, we're going to look at diffusion. So just the definition of passive transport. This is the process in which substances move into or out of the cell without the use of energy. Key word, no energy being used. The substances are going to move based on the concentration. And we're going to call this a concentration gradient. Now, this definition is a little wordy. It says the difference in the number of molecules per volume of substance between a high and a low concentration region. What this is saying is you've got one area that has a lot of molecules, we call that high concentration. We've got another area with very few molecules, we call that low concentration. And that causes this gradient, or this difference between the high and the low. And what you're going to see is that the molecules are going to move from the high part to the low part, trying to even them out, give it that equilibrium. So once again, we have 3 types of passive transport: diffusion, osmosis, and then facilitated diffusion. We're going to focus on diffusion now. We'll look at osmosis on Tuesday, and we'll look at facilitated diffusion later in the week. So diffusion. The actual definition of this is going to be the movement of molecules down a concentration gradient. And when I say down, once again, that's meaning from a high concentration to a low concentration. Some examples, and I'll show some pictures on the next slide. Spraying air freshener, making tea, making Kool-Aid. So here are my pictures. I have a fart, I have tea, I have cooked bacon, I have air fresheners. Think about somebody who's sitting next to you. If that person were to fart, it's really concentrated right by them, and over time it's going to spread throughout the room. It's moving from a high concentration to a low concentration. If you put a tea bag in a cup of water, the tea will start to spread out throughout the cup. It goes from a high concentration at the bag to a low concentration into the surrounding area of the water until it all becomes even. Cooked bacon. If somebody is cooking bacon in your kitchen, after a little bit of time, that smell starts to go throughout the entire house. High concentration in the kitchen, low concentration everywhere else. And lastly, with an air freshener. If I stand up at the front of the room and I spray an air freshener, over time it's going to slowly move to the back of the room. High concentration up front, low concentration in the back. It's going to diffuse or move from the high to the low. So this is what it looks like with the actual molecules. The red dots are our solute, our water, or the blue is going to be our solvent, and then when they all mix together we call that a solution. This should be a review from last year. The solute is going to be the stuff that gets dissolved, or the molecules that are going to be essentially moving. The solvent is going to be the stuff that is helping to dissolve or move that substance. And then the solution is going to be when it's mixed. And so when we have diffusion, we have a high concentration that eventually moves into the low concentration. And when it's done moving, we have an equal amount. We have an equilibrium, or homeostasis has been reached. This little bar here in the middle with the holes, that is our semi-permeable membrane. So only some things can move in and out of this. If this was in a cell, it would determine what moves in and out. But once again, it's in diffusion because no energy is being used. It's always going to move from this high concentration to the low concentration. So I have a quiz for you. I want you to think about it, write your answers down. We will go over this on Monday. I want you to tell me, in lemonade, and let's say this is just like the powder kind of lemonade, what is my solute and my solvent? So I've got my powder, I have my water, I pour it, I mix it. I want you to tell me which one's my solute, which one's my solvent. Same thing with food coloring and water. If I have a cup of water and I have my food coloring, I want you to tell me, if I put food coloring into that water, which one's going to be my solute and which one is going to be my solvent. We will talk about this on Monday. All right, so we're talking about this stuff moving from a high concentration to a low concentration, but how do we know what determines how quickly it does this? There are a couple things. One, the gradient steepness. So this means that there's a bigger difference in concentration. So if I've got an area with a lot of molecules in it, but then an area with very little molecules, I'll have a big gradient, and so that means the molecules will move faster because it's steeper. If my molecules are small compared to bigger ones, then they will also move faster. Kind of think if you have like a big sumo wrestler and then you have a small runner, if they are trying to get to the same place at the same time, which one is going to move faster? Hopefully the smaller guy is going to be able to move quicker than the sumo wrestler. He's a bigger molecule, or a sumo wrestler is bigger than the smaller person. So the smaller one can move much faster. Lastly, the temperature. Anytime you heat something up, those molecules are going to move faster. They're going to make more collisions. It's going to allow things to move faster. So the higher the heat, the faster it's going to move. If you cool something down, they don't move as fast. It's going to be much harder for them to kind of spread through things. We are going to do a kind of mini lab in class on Monday looking at these factors that actually affect how this diffusion or these factors and how it will change how quickly or how slowly diffusion happens. And we're going to do something similar to this, not exactly, but I want you to kind of think about this. If I have 3 beakers and one has hot water, one has warm water, and one has cold water in it, what's going to happen when I put food coloring in each of those? And if you look at beaker number 3, that should say 10. That 2 is a typo because we want to have the same amount of food coloring in each one. But if I have hot, lukewarm, and cold, which one do you think is going to diffuse the fastest in? And then the other way we're going to look at it too is if we have all of the same temperature water, let's say it's lukewarm, but we put different concentrations of food coloring in, once again, which one is going to diffuse faster or move faster? So think about that. We'll talk about it on Monday, and then we're going to do a in-class kind of mini lab with this. This is all for the notes. We will look at osmosis on Tuesday and be prepared to do a mini lab on Monday.